Hello, everyone, and welcome to Lunch Break Science. I'm Ariel Johnson from the Leakey Foundation, a nonprofit dedicated to supporting human evolution research and sharing discoveries in programs like this. We'd like to thank the Ann and Gordon Getty Foundation and Camilla and George Smith, whose generous support made this episode of Lunch Break Science possible. March is Women's History Month, and it's been fun to reflect on the amazing women I've had the opportunity to feature on Lunch Break Science. The Leakey Foundation helps ignite and sustain women's careers through our grants and programs. In honor of Women's History Month, Dr. Kelly Stewart and Dr. Sandy Harcourt, along with the Ann and Gordon Getty Foundation, have sponsored a quadruple match donation challenge. We only need $1,400 more to reach our match goal, so I thought I'd reach out to my Lunch Break Science friends and ask for some help. How it works is you can click on the link we've shared in the chat, or you can visit us at leakeyfoundation.org/donate, and all donations up to $1,400, the first $1,400 worth of donations will be quadruple matched. So if you give $25, it'll be $100 worth of impact. If you give $250, that'll be $1,000 of impact. So we're just really, really grateful for all of the support you've given us and continue to give us. Here with us today is Leakey Foundation grant recipient Ashley Hammond. Ashley, thank you so much for being here.
Oh, I'm so delighted to join you, Ariel. Of course, I, like all of your lunchtime guests, wish that we could be there in person, but it's really delightful to join you.
Well, we're delighted to have you. Ashley is joining us from New York, where she is Assistant Curator of Biological Anthropology at the American Museum of Natural History. She is also Assistant Professor, not Curator. You're not a curator two places. Assistant Professor of Anthropology at the Richard Gilder Graduate School. And Ashley uses the fossil record to understand the origins of different types of locomotor movement of our human and ape ancestors and human and apes, especially bipedalism. This research takes her to East Turkana in northern Kenya, where her work focuses on Pliocene and Pleistocene hominid evolution and paleoecology. She is also co-director of the Southern Kenyan Rift Project around the site of Lemedongo, where her work focuses on the late Miocene. Today, she'll be discussing how fossils, especially the pelvis, help in reconstructing the evolution of locomotion in apes and humans. Before we hear from Ashley, though, if you're joining us live on Leakey Foundation Live, YouTube, Facebook, or Twitter, you can submit questions at any time for Ashley in the chat, and she will be answering those questions at the end of the episode. The earlier you get those questions in, the more likely Ashley will be able to answer them. Ashley, what does the pelvis have to do with the way that apes and humans move?
Oh, it has a lot to do with it. So for those of you at home, the pelvis is the bony structure where your hip bones are. And we tend to think about bipedal locomotion in terms of the legs moving, but the pelvis is a really important part of the equation. So the pelvis is where some of the main locomotor musculature attaches. And it's particularly important for the muscles that are moving the thigh and lower limb. And we actually see that it is completely transformed in bipedal hominins as compared to apes. And in addition to locomotor behavior, the pelvis can tell us all kinds of information. And I'll talk more about this in my presentation, but it can tell us about trunk morphology. It can tell us about obstetric and biomechanical requirements. So when we find a fossil pelvis, it is a goldmine of information.
You've been working in East Turkana for a number of years now. What's going on there, and what is it like to work there?
So East Turkana, I guess I should tell you a little bit about where it is. It's called East Turkana because it's east of Lake Turkana. And it is this really dry and barren kind of badlands type of landscape. And I have told people it's kind of like you're working on the surface of Mars. It's really dry. And to give you a sense of scale, East Turkana, the fossil-bearing deposits are about the size of the state of New Jersey. So it's a really expansive, big place. Now, I got my start working in East Turkana in 2009 when I was invited out by Leakey, and I joined her crew that summer. And then I really increased my participation in fieldwork in East Turkana starting around 2015 or 2016. And I started going out with the Kubifora Field School and the National Museums of Kenya. And so there is so much great research happening in Turkana. There's a number of different research teams that work throughout the region, not just on the east side of the lake, but all around the lake. And you know, just with the National Museums of Kenya and Kubifora Field School alone, we have built these really big research programs in paleontology, archaeology, geology, even human biology. And they're all very complementary and intersect on the same themes and help inform one another. So it's a really fruitful collaboration. Now, one of the things that I particularly like about working in East Turkana is that, well, it's a very fossiliferous location. So there's tons and tons of fossils that are just kind of eroding out of the ground. And the time period that is really fossiliferous is about 1.8 to 2 million years old. So that's a really interesting time period in human evolution, and in East Turkana in particular. So there's a number of hominins that are present on the landscape in East Turkana sometime between 1.8 and 2 million years ago. So we've got Paranthropus boisei, and Paranthropus boisei, I have kind of described this species to people as a lean, mean eating machine. It had big, massive muscular attachments for chewing muscles on the head, and these really massive premolars and molars for chewing probably pretty abrasive foods. We also have Homo habilis, and Homo habilis is one of the earliest species of Homo and kind of a transitional species of Homo. We also have Homo rudolfensis, which we know less about this species. We don't really know what it looks like below the neck, but it seems to be there as well. And then we also have some of the earliest evidence for Homo erectus in East Turkana during this period. So we've got at least 4 hominin species on the landscape, and it's really an exciting place to work.
In comparison to when you first started going out in the field to now, has the amount of time you've spent in the field changed?
Sure. So looking back on my career, when I was a graduate student, I was really trying to finish my dissertation. So I wasn't going out nearly as much as I did later. And my first field experience, as I said, well, my first non-field school experience, I should say, my first time joining a field crew was with Leakey in 2009. And I went out for pretty much the entire summer with Meeve's crew that year. And I had the opportunity to do field work a number of other occasions as a PhD student, but it really picked up when I was a postdoc. And when I was a postdoc, I started going out anywhere from 1 to 3 months a year in the field. So that's a long time to be living in a tent with no plumbing and cut off from your family and loved ones. And now now that I'm in a tenure track position, I have a lot more institutional obligations and so forth. So I have scaled back the fieldwork to, I would say, about 5 weeks total a year. Still a good chunk of time.
Yeah, it really is. You recently began working at the late Miocene field sites around Lamodongo with Dr. Chalomanthi of the National Museum of Kenya. Why did you start this project?
Oh, well, the late Miocene is a really important time period for human evolution. So I have to give credit to Leslie Alusco. She was the person who really put this site on the map about 20 years ago. And Leslie has since moved on to other field sites, but Chalo and I we picked up the fieldwork at this site in 2019, and we've been going there with the hopes of finding late Miocene apes or hominins. And, in terms of, as you mentioned at the start, what I'm really interested in is the evolution of locomotor behaviors and bipedal origins. And so if you really want to know what's happening that led up to the advent of bipedality, the late Miocene is the time period that you need to be working in. So we know that there's tons of locomotor diversity in fossil apes, but we don't really know yet what was going on in the late Miocene African ape fossil record. There's a really poor fossil record for this time period. So we're hoping to help kind of flesh in some of the time period around 6 million years.
What are the different types of questions you look at this site opposed to the East Turkana locations?
Well, the two sites sample very different time periods. So Lamodongo and the sites around it are all dated to right around 6 million years. So that snapshot in time can help tell us about origins of bipedality and the very earliest stages of human evolution. And it can also tell us quite a lot about the paleo environment in which earliest human evolution was happening. As compared to East Turkana, we have a very different situation there. There are plenty of hominins on the landscape. We have evidence of stone tools and so forth. So the kinds of questions we ask or can be answered by the paleontological record are a little different.
You're a curator at the American Museum of Natural History, which I feel like it's been too long to congratulate you, but I still feel like I want to congratulate you. Congratulations. So how does being a curator differ from being at a university?
That's a good question. I think people think my job is quite different than it really is. My day-to-day job is actually quite similar to any tenure track assistant professor position. So a combination of research, teaching, and administrative duties. So obviously a big part of my job is to conduct research and field-based research in particular. We also have a graduate program at the museum, so I'm involved in graduate education. But we have, in terms of my day-to-day activities, some of the undergraduate teaching is substituted for other kinds of activities. So I have unique opportunities to engage in public programming, outreach, some exhibition consulting, and of course, I curate collections.
Yeah. Well, so could you tell us a little bit more about being a curator or the curation part of your curatorship?
So I am the biological anthropology curator. So I curate the biological anthropology collections. And one of the greatest resources that I have is access as a curator to all of the incredible hominin casts that are stored just down the hall from my office. So I think this is actually the biggest perk of my job, is that anytime I want to compare something that I'm working on, I can just zip down the hall and pull out an orangutan, or I can go down the other direction down the hall and open up a drawer full of Homo erectus casts. So it's having the opportunity to work with collections is really an exciting part of my job.
And you also have some influence over exhibits as well.
A little bit. So I haven't begun doing much with the exhibition work because it's incredibly time consuming. But yes, I certainly am involved in some of the exhibition discussions and so forth.
It's got to be really, really fascinating to think about. How did you become interested in pursuing science as a career?
So I got interested in anthropology when I was really young. So I think my first clear memory of kind of discovering paleoanthropology, though, I was probably about 5 years old, and my parents were National Geographic subscribers, and they had an old copy of it was a 1985 issue of all about human evolution. And I think what initially drew me to it was on the cover, it had a holographic Tong child skull. And so as a child, it was pretty cool, but it also had these pages that would just pull out and you could see what was imagined, the course of human evolution illustrated through time. And it was something that really cemented it was a treasured object for me. I still actually have my copy of that issue in my office. And when I went to college as an undergrad, I didn't really think about it, but as something other than an interest. But I realized when I was in college that it could actually be a career path. And I majored in anthropology, and I participated in a paleoanthropology field school in South Africa. And I think that was around 2005. And we excavated at Swartkrans. And, that really almost sealed the deal for me. I knew that I was interested in paleoanthropology, but after doing the field school, I knew I wanted to do field-based research somewhere in Africa.
Do you have any advice for those who are watching today who are interested in pursuing biological anthropology as a career?
So one of the most important things it was a piece of advice given to me when I was finishing up my undergraduate degree. And I was encouraged to take human gross anatomy. And I think for people who want to become paleoanthropologists or functional morphologists and work with bones or fossil bones, anatomy training is absolutely essential. In order to really understand the fossils and what you're looking at, you have to really know your anatomy in and out. So I took human gross anatomy my very first semester. It was the very first graduate course I took when I was a master's student. I taught it over the years, and I actually ended up completing a PhD in anatomy with Carol Ward at the University of Missouri. And it's been something that I credit with the anatomy training and exposure to really knowing what I'm looking at with fossils.
Speaking of fossils, do you have a favorite fossil?
So this is a tough question. It's almost like you're asking me to choose my favorite child.
Oh, yeah.
But I think one of my favorite fossils of all time is a very poorly known pelvis and femur. It's called KNMER 5881. And so you're looking at just a little piece of a femur shaft that was found in 1980. And the reason why I love this fossil is because it's a detective story, and I was part of it. So in 1980, this little piece of femur shaft was found, and it was suggested to be Homo habilis-like in cross-section. And then in 2009, when I was out with Neve Leakey's team, we found more of the femur shaft and part of the pelvis at the exact same spot where this bit of femur had been found in 1980. And back in Nairobi at the National Museums of Kenya, we discovered that you could actually perfectly conjoin the piece from 1980 with one of the new pieces that was found in 2009. So basically, fossils found 30 years apart that clearly belong to the same individual. And so Another exciting thing about this fossil is that this pelvis looks really different from the other pelvises that are known from the same time period. So it potentially gives us some insight into what the pelvis in early Homo looked like. And that's really cool. So we published this, actually Carol Ward was the lead author of this, and it was published in 2015 in the Journal of Human Evolution.
We'll have to, I wish we should have had a link for that article, because I want to read that now. But now let's turn the virtual floor over to Ashley and hear more about the evolution of the pelvis and origin of bipedalism.
Okay, everyone. So thank you for tuning in. I know this is lunchtime for those of you on the West Coast, or maybe post-lunch nap time for those of you on the East Coast. When I start off talking about the pelvis and hips, I think I like to start and step back a little bit and think about why it's important first. So, I always want to ask, okay, what is it that's unusual about humans? Okay, so when we think about humans have relatively large brains. We make and use tools. We have symbolic expression. We do all sorts of really weird things, like watch seminars over Zoom at lunchtime, right? But none of these are present in the earliest hominins. And when we go to the fossil record and look for early hominins, what stands out are signs of bipedality in the skeleton. Bipedality is one of the defining characteristics for our lineage. Now, when we go to the early hominin fossil record, so you're looking at Australopithecus afarensis, this is Lucy's species, we see evidence of bipedality throughout the skeleton, right? So we see it in the bicondylar angle of Lucy's femur. So her knee was positioned kind of under the midline of her body. We don't have this preserved in Lucy, but we know from other Australopithecus afarensis fossils that they had an arched foot, a fairly stiff foot with a big toe that was adducted or in line with the other toes. The head was positioned over the vertebral cord, or over the vertebral column. And there was a short and wide pelvis. And we'll talk more about some of the details of the pelvis in just a second. But we see basically evidence of bipedality from head to toe, right? When we look at slightly earlier hominins, the evidence for bipedality is a little bit more nuanced. So when we go to an early hominin, or even earlier hominin, like Ardipithecus ramidus, Ardi is dated to about 4.4 million years ago. There's still some evidence that this was a species getting up into the trees quite a bit, right? So Ardi has fairly long hands and arms. The feet have big toes that were grasping. So there's some evidence that Ardi wasn't quite as adapted for bipedality, or maybe less committed to bipedality. But the pelvis of Ardi is really strongly suggestive that it was a bipedal hominin. So it seems like the pelvis is one of the earliest parts of the anatomy that really starts to track locomotor behavior. Now, the pelvis also tells us about a lot of other things. So in addition to derived features related to locomotion, we see differences in modern humans. In recent humans, we see differences in the pelvis related to ancestry and where they lived relative to the equator. So we tend to see slightly broader pelvises in people from Arctic climates as compared to people who live in equatorial regions. We also see differences in male and female pelvic shape. And of course, this relates to the fact that women have to gestate and then give birth to large brain babies. Now, when we look at the hominin pelvic plan overall, there are a number of features in common. Okay, so generally speaking, all hominins have a number of features to the pelvis. So they tend to have a really large anterior inferior iliac spine. This is an important site of muscular attachment for some of the anterior thigh muscles. We also see a particular rotation to the ilium that we don't see in other apes, fossil or living species of ape. So the ilium tends to be rotated from facing straight on the back to a little bit more rotated around the sides to form more like a bowl shape. We also see a short distance between the hip joint and the sacroiliac joint. So this is part of the overall reorganization that happens in the pelvis, but the distance between these two joints tends to get a bit shorter in hominins. But then we also see differences that are more specific to the human pelvis. So we see a number of things. So the upper part of the hip bones, the ilia, these don't flare quite so far to the side in modern humans. We also see a mound of bone that's on the ilium called the iliac pillar. This tends to not be very strongly developed in humans as compared to some of the earlier hominins. The pubic ramus is fairly short in recent humans. And then the ischium, the butt bone, is also kind of tucked in a little bit differently. So even though there are these features that characterize hominins overall, there are a number of features which are very particular to humans. So in my mind, as I've been working on the pelvis over a number of years, it seems like there are two big transitions that happen within hominin pelvic evolution. So the very first one is associated with going from an ape-like pelvis to an early hominin pelvis. So a key question on that side is, what did the pelvis of late Miocene fossil apes look like? You know, what kind of pelvic shape did the earliest hominins evolve from? Another big question is what changes sort of led to the human-like pelvic form? So another big question that's unanswered is, what did the pelvis of earliest Homo look like? Now, because today is a relatively short talk, I'm going to talk about just one of these questions. So what did the pelvis of late Miocene fossil apes look like? And what can that tell us about early hominins? So what you're looking at right now is a collection of the Miocene ape hip bones. So these are what the hips look like for fossil apes. This is all we have for the fossil ape record for hips. There are lots and lots of species of fossil apes. Some of the earliest ones tend to have hips that look more monkey-like. So for instance, the two on the left-hand side of the screen, Ikembo and Shiphopithecus. These are often described as having a very monkey-like appearance. Overall, none of these pelvises look exactly like a hominin though, except for perhaps this one on the far right side. So this is a pelvis of something called Oreopithecus. And Oreopithecus is a fossil ape from Italy, dated to about 6 million years ago. And you can see that it has kind of a short, broad shape, kind of like a hominin. So over the years, I wanted to take another look at Oreopithecus, because what you may not have appreciated in that last image, but perhaps you can see here, is that Oreopithecus is a almost two-dimensional fossil. It's really smushed flat as a pancake. So some of what we saw in that slab on the last slide might just be the result of how it was preserved, so deformation. So in 2017, I went to Florence, where this material is housed. Definitely not a bad place to do research, I have to say. And I worked on the original fossils. Okay, so this is what that fossil looks like in the slab. And as I said, it's really almost a two-dimensional fossil. It's very, very flattened. So in looking at the pelvis, it turns out that the pelvis is actually composed of quite a number of fragments. It's not just one piece. So working through it, we identified a number of different pieces to the pelvis. And a lot of what we were seeing in that slab was actually the result of how the fossil had been compressed during fossilization. And so some of the big conclusions that we made based on study of some of the individual elements was that, in fact, Oreopithecus differs from hominins in a number of key ways. So it does not have that protruding anterior inferior iliac spine, which is a key site of muscular attachment for being a biped. It also did not have a particularly short distance between the hip joint and the sacroiliac joint. And the ilium was not rotated like a modern human or even a fossil hominin. So there were a number of features that looked nothing at all like a hominin. But this was not exactly like a living species of ape either. It doesn't look exactly like a chimpanzee or a gorilla. So having these fossil apes like Oreopithecus these are really great because it helps us calibrate our expectations about what the ancestral apes might have actually looked like. And with that in mind, that is why my colleague Chalo Manthi and I have undertaken new fieldwork in late Miocene sites in Kenya. So the reason why we want to work these late Miocene sites is because this is the critical time period when apes and hominins were diverging, right? So we want to find some of the earliest evidence for hominins or perhaps even an African ape fossil at these sites. So the site that you're looking at is called Lemudongo, and you can see there's little specks of people on those outcrops. That was our field crew working in 2019. And although we haven't found any apes or hominins at this site yet, there's a lot of potential at this site. So you can see here, these are some of the fossils that we've been finding, and they have really excellent preservation. And the fossils that we're finding are also characteristic of a fairly wooded or closed environment, as opposed to a more open grassland type environment. So this is really promising, we think, because the earliest hominins are thought to have evolved, hominins like Ardipithecus are thought to have evolved in a slightly more closed environment, a more wooded environment. And this is our field crew. I just want to go ahead and acknowledge some of my colleagues here, Dr. Chalo Manthi in the center, as well as Aaron DiMaggio of Penn State. We had hoped to go back out again in 2020, but unfortunately, because of the pandemic, we had to cancel our field season. But please stay tuned. We hope to be back with Leakey and present more about future field seasons once we get out. And with that, I would like to thank all of these institutions, people, and funding agencies.
Thank you so much, Ashley. That was a great talk. We'll now be taking some questions from the audience. If you haven't gotten those questions in the chat, be sure to get those questions in right now. We have an initial question from our website, which will be coming shortly. How often do you find a piece of pelvis that you are able to study?
That's a good question. So the pelvis is regrettably not preserved very frequently. And when it is, it's often kind of banged up. So the pelvis is a very irregular shaped bone with a lot of edges and corners and thin parts to it. So unfortunately, it tends to not be preserved very well or very often. But when we do find a pelvis, as I said before, it's just a gold mine of information because there's so much that it can tell you about all kinds of aspects of an individual's biology.
Is there a particular part of the pelvis that is better, more
likely to preserve?
Yeah, more likely to preserve. Yeah.
Yeah. So I've been involved. In all the years that I've been working in East Turkana, we have found a hominin pelvis on 2 occasions, and on both occasions, they have preserved parts of the sacroiliac joint and the ilium towards the hip bone or towards the hip joint. So I think those are the parts of the bone that tend to be a little bit thicker, and I think that's why they preserve. Unfortunately, the parts near the front of the pelvis, like the pubic bones, are very thin and just very prone to being broken or I think sometimes eaten.
Are they very clear that they're, human or hominin, as opposed to other animals from fragments? Is it a little harder, or in comparison to like other?
So hominins have such a distinctive pelvic shape that yes, it is. When you find a piece of hominin pelvis, it's usually very obvious that you found a piece of hominin pelvis. There's really nothing else, for instance, that has a really well-developed sciatic notch, greater sciatic notch. So usually to a trained eye, it's pretty apparent.
Let's take our next question from the audience. How do you decide where to excavate?
So typically in East Africa, often the first step before excavation is actually to do ground surveys. And so what this entails is we identify an area that we think appears to be fossiliferous, and then we will walk the surface and look for locations where it seems like we're finding really nice vertebrate fossils coming out of the ground. And once we have identified a focal area where we want to focus, then we might excavate. Certainly if we find what appears to be a hominin or a hominin skeleton coming out of the ground, we would excavate. But usually the first step is to do those ground surveys. And I think my piece of advice for those is you always want to be friends with a really good geologist, because the geologists know where to point you to in terms of where to do your survey work.
It's a second direct good piece of advice. Okay, let's take our all right. There's like your talk was already loaded with great advice, but okay, let's take the next question from the audience. How could you speak further on how COVID has affected your research and the activities of your colleagues?
Yeah, I think COVID has unfortunately impacted us all in different ways, of course, but I think it's certainly prevented most field teams from returning to their sites. So I think as long as COVID remains a threat, we can't in good conscience return to our field sites where we might expose rural communities.
Yeah.
Of course, we also are concerned about our American team members, our students, and so forth. So first and foremost, safety is, I think, at everyone's mind, and that's made a lot of our decisions over this last year. But we also see COVID impacting us in our lab work, in our day-to-day activities. It's, I think most of us are cut off from our offices and may even have budgetary freezes and so forth. So it's certainly impacting all of us. And I imagine that some of the most profound impacts will be felt in the next 2 or 3 years.
We have another question. Cheryl asks, how are you helping to inspire, teach, train the next generation of women paleo scholars in Kenya?
Oh, well, that's great. So a big part of, as I mentioned before, I participate in the Kubifora Field School. And one of our main training missions is to train not only the next generation of women scholars, but Kenyan scholars in particular, because we work in Kenya. So we always reserve about 10% of our student capacity is for fellowships to Kenyan students to come at no cost to them and to train them in Kenyan field science so that we can help give back to the community. So we've been doing this over many, many years, and we've trained at this point, I would say dozens of Kenyan and East African young scientists.
We have actually we're getting quite a few questions, so let's get on to the next question. Are there any fossils found in Africa that seem to connect with Rudopithecus, or do you think Rudopithecus were also in Africa or something similar?
Well, that's a good question. So for those of you at home who don't know what Rudopithecus is, it's a fossil ape from technically the late Miocene, although kind of the early late Miocene in Hungary. And it is one of the later lived species of apes in Europe. And we don't have anything that looks like Rudopithecus in Africa that we have found yet. We do have some fossil apes that are about the same age. So Rudopithecus is about 10 million years old. And we do have a few fragments of ape from Eastern Africa at that same time period. So we have Curator and Nicalipithecus, which are right now only known from dental material. So we really can't say if they look like Rudopithecus yet. The teeth don't look exactly like Rudopithecus, but we don't know what their skeletons look like. So it's possible, totally possible.
Let's have our next question. What can a fossil pelvis tell you about childbirth?
So we can get a sense of sort of some of the major changes that have happened in the hominin lineage that we think might be related to childbirth. So certainly it seems like over time, the shape of the obstetric canal changes. So the obstetric canal is the large hole that the baby has to pass through as it exits out of the pelvis and then out of the mom's body. And in early hominins, it tends to be a more oval shape. And then over time, it gets to be more rounded. And we think there was probably some selective pressure to make the shape a little different to facilitate large brained babies going through. And then there's also occasionally you can find some evidence of childbirth based on some scarring that will occasionally develop on the pelvis as a result of childbirth.
We're getting quite a few questions. Anna Marie asks, would you say that the evolution of the pelvis has favored bipedal locomotion, but to the detriment of childbirth? Is the actual strategic in any way for the evolution of our genus?
So this is still a kind of hotly debated topic. What was a bigger pressure on pelvic shape evolution through time? Was it locomotion or was it childbirth? And I can't say that I have entirely made up my mind one way or the other. I think they were both really important pressures. So unfortunately, I have to say this one I think is still unresolved.
Jury's still out.
Jury's still out.
But that makes it more fun. Still have questions.
This is why we need to find more fossils.
Yes, exactly. So let's look at the next question. James Cardy asks, have changes in our gestation time periods evolved in humans along with those long-term changes in locomotor evolution? And if so, how was this found out?
So we're still not sure exactly what gestation time would have looked like in these fossil species. It's really hard to know. So I can imagine certain ways that you might look at tooth development on a micro scale in a fetus or some very young individual. Maybe you could identify exactly how long it had been developing, but we just simply don't have the tools to do this right now. We don't have the fossils to do it, and I think it would be very hard to come up with those estimates for gestation time. So unfortunately, I have to pass on this one. I don't think we know yet.
Our next question. Have you encountered any difficulties as a woman working in science with regards to career advancement?
Well, there's always, I think, challenges. So certainly being female, paleoanthropology has for many, many years been a very male-dominated field. But that's been changing. There are a number of incredible female scientists from the generation before me that paved the way for people like me. And I think if you look at my peer group and even younger, there's lots and lots of incredible women doing incredible things. So I think it has certainly been a bit of a barrier in the past, but I think the times are changing.
We have, I think, another question from from the same person. There are many travel advisories regarding the Turkana region in Kenya due to safety concerns. Has this impacted research in the area pre-COVID and in the future?
So certainly there are travel advisories to many places, and there are some to Turkana. I think my biggest concern working in Turkana relates mostly to things that can be avoided, like malaria. But in terms of it being a concern, perhaps to political instability or some kind of violence or something like that, that's not really a concern at all. We work in such a remote place that we very rarely encounter other people or points to be concerned about. So I think the biggest concern for me usually is making sure that I use bug spray and don't get sunstroke, drink a lot of water.
Our last question of today will be, knowing that it's Women's History Month, and there are so many amazing women in the field of anthropology, who are the women who've inspired you?
Well, as I said, there are many incredible women doing things now. But I think in terms of people who inspired me when I was, especially at earlier career stages. So first and foremost, I have to say Carol Ward. She was my dissertation advisor, and she's a brilliant scientist, and no one knows hominin postcrania the way that Carol does. So she's absolutely one of the best people to work with. I also have found Kay Berensmeyer to be very inspiring. Kay has worked in East Turkana for many years. She's been just a luminary in developing taphonomy and finding better resolution on the geology and paleoanthropology more broadly in Turkana. So she's really an inspiring figure. Leslea Lusco, who, as I mentioned before, I, in some ways, inherited the Lemudongo field project from her. She was my predecessor at the site, and I couldn't have asked for a more generous or well-organized predecessor. She really paved the way for me. And then I think, lastly, I would say Niamh Leakey, who allowed me to join her field crew very early on when I was a PhD student and get my feet wet in East Turkana paleoanthropology. And yeah, those four ladies really have made a lot possible for me.
Well, it was a quadruple answer there for that question, which kind of leads us back to our quadruple match. Again, we would love if all you watching would help us fund some amazing research and programs. Thanks to Dr. Kelly Stewart and Dr. Sandy Harcourt and the Anne and Gordon Getty Foundation. We have the quadruple match going on. You can visit us at leakeyfoundation.org to learn more about Ashley, as well as how you can help us support research. We've shared a link in the chat. On April 1st, in 2 weeks, we meet Leakey Foundation scientist Kate McGrath and learn how the teeth of our ancestors can tell us about their life histories. Be sure to subscribe to Leakey Foundation's YouTube channel, follow us on Facebook and Twitter, or sign up for our newsletter to be the first to hear about upcoming episodes, as well as groundbreaking discoveries in human evolution research. Still hungry for science and can't wait till April 1st? Check out the Leakey Foundation's award-winning podcast, Origin Stories, available wherever you listen to podcasts. Lunch Break Science is made possible by the generous support of the Anne and Gordon Getty Foundation and Camilla and George Smith. Thank you all for taking a break from your day and feeding your brain with the Leakey Foundation. Until next time, stay hungry for knowledge.